This weekend, I was super fortunate to have an epic adventure. I went up into the Reiwa Mountains with the ski team, and of course Elise was there, and we ran in the Reiwas for 6 hours. It was an incredible run. Of course, left us famished at the end, and I continued to be famished all the way through last night, and I decided let's cook an epic dinner. So we cooked these incredible latkes with sweet potatoes and radishes and all manner of greens from Acre Student Farm, and I was sitting back after that, relaxing just a little bit, and thinking about how my body was passing into rest and digest, and how the glucose levels in my blood were very, very high. And recall that the first of our 3 metabolically irreversible steps, catalyzed by hexokinase, has multiple isozymes. Remember that glucokinase is one of those, and glucokinase has a Km that is very, very high. So it doesn't really get going until blood glucose levels get really high. So my glucokinase enzyme was upregulated because my blood glucose levels were so high. And so my body was saying, you know what? It's a good idea to store away some of this excess glucose in these times of plenty for that next 6-hour run when Rachel is going to tap into and deplete all of her glycogen. So my liver began to focus, instead of on breakdown of glucose, it began to focus on storing away that glucose for a time of need. That is, glycogen synthesis became upregulated. Now there is a very famous and important enzyme involved in glycogen synthesis. Notice that glucose 1-phosphate is the first molecule formed along this pathway. It then serves as the building block, the monomer, to make the polymeric polysaccharide glycogen. And the famous enzyme here is glycogen synthase, very fittingly named. Glycogen synthase is heavily regulated to regulate whether or not glycogen should be synthesized or broken down. Now, the enzyme that is equally as famous that breaks down the glycogen, that would have been the one going to town during my 6-hour run. This is glycogen phosphorylase. It's going to release the monomeric subunit of glycogen to make glucose during those times when the liver needs to serve as the food bank and ship glucose out to other tissues. Which, remember, that means that the liver is one of those very special tissues that has the ability to bypass this reaction in which glucose is converted to glucose 6-phosphate, metabolically irreversible. So there's a bypass reaction, an enzyme that will take glucose 6-phosphate back to glucose that will then allow it to ship glucose out to the other tissues. So when I'm running, my muscle tissues will get the glucose that derived from my storage within my liver food bank. And I'm always feeling very grateful for that. Now, it makes so much sense that glycogen synthase and glycogen phosphorylase are reciprocally regulated. That is to say, it would be stupid to synthesize glycogen while you're still breaking it down. So when glycogen synthase is turned on, glycogen phosphorylase is turned off. Hopefully you've never had a relationship like that. But these are very reciprocally regulated. And we want to dive in and talk more about this process of both glycogen synthesis and glycogen breakdown. As we dive into chapter 12, then, we have zoomed in to the upper left-hand corner of our prior big picture, and we're looking specifically at that corner whereby glycogen is synthesized and broken down reciprocally. And the term that we give to glycogen degradation is sometimes a special term called glycogen lysis, which makes a lot of sense, the lysis of glycogen. And shortly we will focus in on the reaction by which glycogen phosphorylase converts glucose to glucose 1-phosphate and glucose 1-phosphate to glucose 6-phosphate and so on forth. But I also want to make sure that we're focusing in on the biosynthesis of glycogen, and that's actually going to be our first consideration. So this is the point at which I was in that rest and digest last night when I was eating the sweet potato latkes. And I want to look mostly at and consider mostly glycogen synthase. So glycogen synthase will be the enzyme of our primary consideration. But that is not to take anything away from UDP glucose pyrophosphorylase. I mean, for one thing, it just has a pretty wickedly cool name. Certainly a mouthful, but also it does do something kind of neat. Remember back to chapter 7 where we talked about UDP glucose, and we talked about this as being a particular metabolite cofactor. I'm going to bring back a picture from chapter 7 whereby we can really look at the details of glucose 1-phosphate getting converted to UDP glucose. And in fact, this is an essential priming or prepping step because the substrate that glycogen synthase recognizes and from which it builds is UDP glucose. So UDP glucose pyrophosphorylase is what will convert glucose 1-phosphate into that UDP glucose. So here's a picture from chapter 7, recalling then the reaction by which UTP, of course, a highly energetic compound, can react with glucose 1-phosphate, and this is going to form UDP glucose, which will then serve as substrate for the glycogen synthase. And now we have UDP glucose, which will serve as the substrate for glycogen synthase. So this is an important reaction and one worthy of our note because the readying step that it plays in getting the substrate ready for glycogen synthase to then build the glycogen molecule is really, really important. And additionally, if we think back through to what was happening, like if I decide I'm going to go out for another 6-hour run, we know that then glycogen synthase will get downregulated, glycogen phosphorylase will get upregulated. And note that glucose 1-phosphate will then be formed again, and phosphoglucomutase will act on glucose 1-phosphate to make glucose 6-phosphate. Now, I want to call your attention to something that is worth our differentiating. And that is that this is a specific metabolically irreversible step by which glycogen phosphorylase acts on glycogen to make glucose 1-phosphate. Well, likewise, this is a set of bypass reactions that are specific and distinct from the glycogen phosphorylase catalyzed reaction. That is different than what's going on here with phosphoglucomutase, which actually is just a near equilibrium step. So if you want to make note of that, I was being a little lazy in the notes there. You can make note that this is a truly near equilibrium step catalyzed by phosphoglucomutase going either direction, whereas here these are two distinct reactions. Likewise, the liver is one of the most wicked awesome tissues, along with the kidney and the pancreas and the small intestine, that has the ability to take glucose 1-phosphate and convert it backwards through a separate and distinct reaction to glucose. So the enzyme that catalyzes this is glucose 6-phosphatase. Glucose 6-phosphatase is only found in particular tissues, and it actually is associated with the endoplasmic reticulum, so it's a very distinct reaction. It's not in any way related to the hexokinase or glucokinase reaction. It is a very distinct reaction that allows your liver to convert glucose 6-phosphate back to glucose so that it can then fuel your other tissues or act as a food bank. So for example, our brain likes to run on glucose, so it's very important that the liver can break down glycogen. Say you've been sitting and studying biochem all day long and you forget to eat, your brain is needing a lot of glucose, right? I'm making you think a lot. But on the other hand, your glycogen is going to need to get broken down to make that glucose because you haven't eaten anything. So then that glucose 6-phosphatase becomes your best friend. This is the enzyme that you are so glad of. It's also the enzyme you're so glad of when you're doing that 6-hour run or whatever sort of exercise you might be doing in which you might be degrading and utilizing those glycogen stores. So that glucose 6-phosphate can allow glucose 6-phosphate, or that glucose 6-phosphatase can allow allow glucose 6-phosphate to convert back to glucose to get shipped off to other tissues to supply them in the style that we know is the style of the liver, being the food bank for other tissues. Now, this is really important and oversimplified on the diagram that we have here, so there is even more that we need to say about all of this. First, though, let's peek at the muscle tissue, because something that is distinct, and notice we don't say that our peripheral muscle tissues don't have this glucose 6-phosphatase, but they do have the ability to synthesize and store glycogen, where we recognize then that the glucose getting transported in via GLUT4, and particularly when insulin is high, and glucose here again getting acted on by hexokinase, being converted to glucose 6-phosphate, and of course being used for energy in glycolysis and the TCA. But during times of plenty, a muscle tissue too, can make glycogen, and our dark, slow twitch, aerobic muscle fibers will be those that store most of the glycogen. And you'll recognize that the enzymes are the same. UDP glucose pyrophosphorylase, making UDP glucose, serving as substrate for glycogen synthase, making glycogen, reciprocally regulated with glycogen phosphorylase, which will be upregulated when your muscle needs to break down that glycogen and utilize it as an energy source. So now we have a sneak peek into these tissues, and at a tissue level, let's think more at an enzyme level.